Welcome to Fluent Daily, the English listening channel for advanced learners at B2 and C1 level. Every episode features a rich, flowing conversation in natural English on a topic that is genuinely worth thinking about: history, psychology, science, philosophy, and more. There are no simplified sentences here, no slow speech, no vocabulary drills. Just real English spoken naturally on ideas that matter. Your job is simple. Listen. Let the language wash over you. You don't need to understand every word. Just stay with the conversation, follow the ideas, and let your English do what it's designed to do: absorb. This is Fluent Daily. Let's get into it.
What if I told you that the entity controlling your anxiety, your mood, and even your cravings right now isn't actually in your head?
Right.
It's in your gut. And here's the truly wild part. Most of it isn't even human.
Yeah, which is just a massive shift in how we think about ourselves.
Exactly. I mean, we have this very top-down, almost architectural expectation of how our biology works. You look at the human body, and the brain sits up there in the skull, encased in solid bone, like an emperor on a throne.
The ultimate command center, right?
Right. We just assume it sends orders down the spinal cord, and the rest of the body just quietly obeys.
Yeah.
It's a very clean, hierarchical way of viewing ourselves.
It is, yeah. It's
Yep.
a very comforting model. The brain speaks, the body listens. For centuries, that was really the foundational paradigm of human biology and medicine.
That's changing now.
Oh, completely, because when you look closely at the neurological and, the microbiological reality of the gut-brain axis, that beautiful, simple hierarchy just completely shatters.
And that is exactly why we are here today. Welcome to your custom deep dive. Today, we are going on an expedition into a biological frontier that is completely redefining how we think about our bodies and our minds.
It really is a frontier.
We're exploring the second brain and the microbiome. And honestly, I am so incredibly pumped to dig into this because the actual verified biology we're going to talk about is somehow even wilder than the science fiction version.
The science is genuinely breathtaking, but we are going to have to tread very carefully today.
Oh, for sure.
Our mission today is to methodically unpack the reality of this internal ecosystem. And to do that, we have to rigorously separate the hard, verified scientific facts from the absolute explosion of commercial wellness hype.
Because the hype is inescapable right now, isn't it?
Yeah.
I mean, you literally cannot walk into a grocery store without seeing a massive wall of brightly colored prebiotic sodas, probiotic powders, and kombuchas.
Yeah, they're everywhere.
All of them promising to, fix your gut, cure your anxiety, and balance your mind, all in one $12 beverage. It's mind-blowing how fast this has bypassed the laboratories and just entered the mainstream marketing machine.
It really is. That commercial enthusiasm has vastly outpaced the actual clinical evidence. So my role today is to be the voice of scientific reason.
I appreciate that. We need it.
We will be carefully examining the methodology and the quality of the evidence at every single step of this deep dive, distinguishing what is robustly established in human biology from what is just commercially motivated speculation. Well, that's going to be our compass.
Okay, let's unpack this. Let's start with the physical hardware. The organ that mainstream medicine essentially ignored for a century. We need to talk about the enteric nervous system, or the ENS.
Right, the actual physical network in the gut.
When I first read these numbers in your notes, I actually had to stop and read them a second time just to make sure I wasn't dropping a zero.
It's a shocking number.
The human gastrointestinal tract contains approximately 500 million neurons. That is more neural tissue than you have in your entire spinal cord.
It is a staggering amount of processing power, and it's located entirely in your abdomen.
500 million.
500 million, yeah. And those 500 million neurons are organized into a network that is so highly sophisticated, it regulates the incredibly complex process of digestion completely independently.
Wait, totally independently?
Yes. It manages the mechanical mixing of food, the secretion of enzymes, the local blood flow, and the absorption of nutrients. And it does all of this without needing to ask the brain for permission.
Wow. I want to talk about how we actually figured this out, because scientists stumbled onto this massive independent brain a really long time ago, right? Two British physiologists, William Bayliss and Ernest Starling, They were doing experiments way back in the 1890s.
Yeah, their work was foundational. They were studying the digestive systems of dogs, and the prevailing theory at the time was that the central nervous system dictated everything. So Bayliss and Starling severed all the nerves connecting the dog's digestive tracts to their brains and spinal cords.
So they basically cut the main power lines.
Exactly. They completely expected digestion to just halt. But instead, they observed that the gut just kept working.
That's crazy.
It really was. It continued to produce localized, coordinated muscle contractions, what we call peristalsis, to move food along perfectly normally.
The gut basically said, I don't need you, to the brain, and just kept doing its job. But despite that massive discovery in the 1890s, the concept of a totally independent nervous system in the gut just got completely sidelined, didn't it?
Unfortunately, yes. The 20th century really became the century of the central nervous system. Mainstream biology became hyper-focused on the brain in the skull.
Right.
The enteric nervous system was viewed as just a primitive, localized reflex network. It wasn't until the late 20th century, largely through the work of Michael Grushon at Columbia University in the 1980s and 90s, that the field was truly revived.
He's the one who coined the term, right?
Yes, he published a book called The Second Brain, because his research proved just how autonomous, complex, and chemically identical the gut's nervous system was to the brain's.
And second brain isn't just a fun, catchy nickname. Like, there is a deep, literal, biological connection between the brain in your head and the brain in your gut. If we look at how a human being actually forms in the womb, they share the exact same origin story.
That's one of my favorite biological facts. During early embryonic development, there is a cluster of cells called the neural crest.
Okay.
This is the very tissue that will eventually form your entire nervous system. As the embryo develops, this shared tissue literally divides.
So it splits in half.
Basically, yes.
Yep.
One portion folds up and develops into the central nervous system, your brain and spinal cord. The other portion migrates all the way down into the developing gastrointestinal tract to become the enteric nervous system.
So they are quite literally born from the exact same biological cloth.
They absolutely are.
Because they come from the same source. They speak the exact same chemical language. And this is the fact that absolutely floors people. When you and I talk about neurotransmitters, specifically something like serotonin, we immediately think of the brain, right?
Of course, serotonin is culturally known as the mood molecule.
Exactly. It's the primary target of SSRI antidepressants like Prozac or Zoloft. But the gut utilizes over 30 different neurotransmitters. And an astonishing 95% of your body's total serotonin is not in your brain.
Right.
It is manufactured in your gut.
To be highly specific about the mechanism there, that serotonin is synthesized by enterochromaffin cells.
Enterochromaffin cells.
Got it. Yeah. These are specialized endocrine cells embedded directly in the mucosal lining of your gastrointestinal tract. That fundamentally shifts our perspective.
I mean, it has to.
The vast majority of a chemical we associate with human happiness and emotional regulation is being pumped out in your intestines.
So, if the gut is manufacturing all this serotonin and it has 500 million neurons, how are these two brains actually talking to each other?
Okay.
Because they aren't just sending wireless signals, right? There's a massive physical cable connecting them.
Yes, the vagus nerve.
The vagus nerve.
It is the 10th cranial nerve, and it is the longest, most complex cranial nerve in the body. It wanders from the brainstem all the way down through the neck and the thorax, branching out to touch the heart, the lungs, and extensively innervating the abdomen.
It's like the main biological highway.
It is the primary biological wire of the gut-brain axis.
Okay, let's unpack this. So, if I'm picturing this whole system like a corporate structure, right? My default assumption is that the brain is this micromanaging CEO sitting in the corner office up in the skull.
Naturally.
And the CEO is firing off a constant stream of orders down the vagus nerve to tell the gut what to do. But when you look at the actual anatomy of the nerve fibers, it's the exact opposite.
The anatomical breakdown is genuinely revelatory. When researchers trace the individual nerve fibers within that massive cable, they find that 80 to 90% of those fibers are afferent.
Meaning what exactly?
That means they are carrying sensory information from the gut up to the brain. Only 10 to 20% are efferent, carrying signals down from the brain.
Okay, so the gut isn't some passive employee just taking orders from the CEO. The gut is acting more like a rogue regional manager who operates entirely on their own, doesn't actually need the CEO to get the daily work done, and spends all day sending massive stacks of reports, memos, and complaints up to the head office.
That captures the dynamic perfectly. The gut is speaking to the brain far, far more than the brain is speaking to the gut.
Which is just wild to think about.
The implication here completely rewrites our medical framework. The gut is not a passive plumbing tube designed solely for digestion and absorption. It is the body's largest sensory organ.
A sensory organ. Wow.
Yes. It is constantly monitoring the chemical, mechanical, and microbial environment inside you and relentlessly transmitting that massive data set up to the central nervous system.
It is incredibly humbling to realize we've been walking around with a half-billion neuron supercomputer in our stomachs this whole time, and we just thought it was a food processor.
It is a profound paradigm shift. But I want to pause here and raise my first major flag of caution for you.
Oh, please do.
The anatomy we just discussed is deeply established fact. We know the 500 million neurons are there. We know the enterochromaffin cells produce 95% of the serotonin. We know the vagus nerve traffic is 80% bottom-up.
Right, all proven hardware.
Exactly. But because this hardware is so impressive, the clinical claims made by the wellness industry have raced light years ahead of what the rigorous research can actually validate. We know the hardware exists, but science is still painstakingly trying to figure out exactly what software it's running.
The software analogy is perfect because it's not just our human cells doing the talking down there. We have to introduce the other half of this equation, which is where things get infinitely more complex. We need to talk about the microbiome, right? The ecosystem within.
Ecosystem is the precise scientific term we should use. When we talk about the microbiome, we are not talking about a few transient bacteria passing through your system.
No, we're talking about 38 trillion microbial cells. Bacteria, archaea, viruses, and fungi. 38 trillion. Just to visualize that, that number actually rivals the total number of human cells in your entire body. From a sheer cellular perspective, you are basically a walking coral reef.
That's a good way to look at it.
You are half microbe.
Biologically, you are a superorganism. And it is crucial to understand that these 38 trillion microbes are not parasites and they aren't just hitchhikers along for the ride.
They're doing real work.
Exactly. This vast microbial community has co-evolved with humans over millions of years. They perform biological functions that we never evolved the capacity to do ourselves, functions that are absolutely essential for our survival.
They are essentially doing heavy lifting that our own genetics outsource to them.
They are the ultimate biological contractors. To give you a sense of their utility, they synthesize essential vitamins, like vitamin K and various B vitamins, that our human cells cannot manufacture.
Wow.
They actively train and calibrate our immune systems from the moment we are born. They metabolize complex plant polysaccharides, dietary fiber that our own human digestive enzymes are completely incapable of breaking down.
Because we literally can't digest fiber on our own.
We can't. And they form a robust physical and chemical barrier along the gut lining that actively fights off pathogenic disease-causing organisms.
So there's security too.
Yes. And vitally for our focus today, they produce a vast array of small molecules and metabolites that enter your systemic blood circulation and influence the function of every other organ system, including your brain.
Here is where I start to get a bit dizzy trying to comprehend how scientists actually study this. Because you would think, okay, if the microbiome is essentially a vital organ system doing all this heavy lifting, we should just be able to look in a medical textbook and see what a normal one looks like. Just like we have a baseline for a healthy liver or a healthy heart.
You would assume so, yes.
But no two people on Earth have the same microbiome.
The sheer variance is staggering. The composition of the gut microbiome varies more between healthy individuals than almost any other biological system in the human body.
Because it's being dynamically shaped by literally everything in your life. I mean, your genetics play a role. Whether you were born vaginally or by C-section sets the initial foundation. Whether you were breastfed or formula-fed.
Your current geographic location, your baseline stress levels, your history of antibiotic use.
Right. And it shifts dynamically based on whether you'd ate an apple or a donut for breakfast or if you stayed up too late last night.
The scale of this variability was actually one of the most sobering revelations of the Human Microbiome Project.
That was the massive National Institutes of Health initiative that launched around 2007, right? Similar to the Human Genome Project.
Yes, exactly. The goal was to systematically map and characterize the human microbiome across different populations to finally establish a baseline. They sequenced the microbes of hundreds of healthy volunteers.
And what did they find?
Immense, unprecedented diversity. There was no single universal healthy microbiome profile. You and I could both be perfectly healthy, asymptomatic individuals, and our microbial profiles could look wildly different.
So here's where it gets really interesting, but also confusing. If I'm a scientist, How do I possibly study what goes wrong with the system when I don't even know what a normal one looks like? If everyone is so different, how do you diagnose a broken ecosystem?
It is the defining methodological hurdle of the entire field. Because we lack a single taxonomic baseline, meaning a master list of exactly who should be there, scientists have had to pivot.
Okay, to what?
They now look for functional patterns. It is much less about taking a roll call of these specific bacterial species and much more about analyzing the genetic potential of the community as a whole. What metabolites are they collectively producing? What genes are active?
And even just taking that roll call seems fraught with technical nightmares.
Oh, the physical reality of gathering the data introduces massive biases. The vast majority of human microbiome research relies on analyzing fecal samples.
Right.
But a fecal sample is essentially the biological exhaust at the very end of the 30-foot digestive tract. The microbial community living in the highly acidic stomach is totally different from the community in the small intestine, which is totally different from the mucosa-associated community clinging to the walls of the colon.
Right. So analyzing a fecal sample to understand the whole gut would be like trying to understand the incredibly complex ecology of the entire Mississippi River by only scooping up a single cup of water at the Delta in New Orleans.
That is a perfect visualization. Furthermore, the molecular methods we use to sequence microbial DNA can introduce their own biases depending on how the DNA is extracted and amplified.
It's just so complex.
And, as we will discuss shortly, we rely heavily on animal models, specifically mice, whose gastrointestinal anatomy and native microbes do not map perfectly onto complex human biology. All of this context is vital because it reminds us to constantly stress the difference between observing a correlation, noticing that two biological things happen at the same time, and actually proving a mechanistic cause.
Okay, let's transition from the anatomy into the action. We've mapped the hardware, the 500 million neurons of the enteric nervous system, the massive vagus nerve. We've introduced the 38 trillion microbial inhabitants. Now we need to look at how these entities are actually communicating in real time. We're getting on the gut-brain axis.
And it is not a single dirt road. It is an integrated, multi-lane superhighway.
We tend to focus just on the vagus nerve because it's the most obvious physical cable. But there are multiple communication pathways firing simultaneously, right? You have the vagus nerve handling the rapid hardwired electrical signals. You have the localized enteric nervous system managing the immediate environment. You have the immune system, and a massive percentage of your body's immune cells are stationed right there in the gut lining, sending out inflammatory cytokines.
Correct.
You have the endocrine system releasing powerful gut hormones like ghrelin, which makes you hungry, and leptin, which makes you full.
Yeah.
And then you have that systemic circulation of the metabolites being pumped out by the microbes themselves.
It is a vast parallel communication network, but the defining feature of this entire system is its bidirectionality.
Meaning it goes both ways.
Yes, the gut is constantly shaping the state of the brain, and the brain is simultaneously shaping the environment of the gut. This isn't just an anatomical curiosity. This bidirectional loop is the core mechanistic explanation for something humans have observed in themselves for centuries: the profound, undeniable link between how our stomachs feel and our psychological state.
If you're listening to this right now, you have absolutely experienced this. We all have. Like the intense nausea that hits you right before you have to stand up and give a public speech.
Classic example.
Or the sudden, urgent loose bowels before a massive high-stakes exam. Or the way your appetite completely vanishes when you receive devastating news. It's like a biological check engine light illuminates in your stomach the second your mind perceives a threat.
Those are acute examples of the brain directly altering gastrointestinal function. But we see this play out chronically in functional gastrointestinal disorders, the most classic example being irritable bowel syndrome, or IBS.
IBS is fascinating because it's so common, yet so medically frustrating.
It is incredibly prevalent. IBS is characterized by chronic abdominal pain, severe bloating, and drastically altered bowel habits, either diarrhea, constipation, or alternating between the two.
Right.
But the defining feature of IBS is that it occurs without any identifiable organic or structural cause. A gastroenterologist can perform a colonoscopy, look directly at the tissue, and find absolutely nothing wrong.
Wow, nothing.
Nothing. There is no visible inflammation, no tumor, no ulcer. The tissue looks perfectly healthy, yet the patient is experiencing debilitating physical distress.
And historically, because doctors couldn't find a physical cause, IBS was often dismissed as just being all in the patient's head, which is a terrible way to frame it, but it recognized that there is a massive psychological component.
For decades, the medical community struggled to interpret that psychological link. But modern research into the gut-brain axis has substantially clarified this bidirectional reality. We now have robust longitudinal data showing that a clinical diagnosis of anxiety or depression can significantly predict the onset of IBS symptoms years later.
Oh, wow. So the anxiety literally precedes the gut issue.
Yes, the troubled mind creates a troubled gut. But conversely, individuals who develop IBS first are at a significantly higher risk of subsequently developing anxiety and depression. The troubled gut creates a troubled mind.
So what does this all mean for the chicken or the egg scenario? If I'm sitting here with a terrible stomach ache and a racing, anxious mind, is my brain misfiring and making my stomach hurt? Or is my inflamed stomach sending panic signals up to my brain?
What's fascinating here is that the clinical reality is the answer is both. It is a continuous, self-reinforcing feedback loop. And to understand how this loop actually functions, we have to look closely at the biological mechanisms of stress.
Okay, walk us through it.
When you experience psychological stress, say you are worried about your job or your finances, your brain initiates a cascade. It activates your autonomic nervous system, specifically the sympathetic fight or flight branch. Simultaneously, it activates the HPA axis.
Let's break down the HPA axis for a second, because it's crucial here. That stands for the hypothalamic pituitary adrenal axis.
Yes, your hypothalamus in the brain senses the stress and releases a hormone called CRH that tells your pituitary gland to release another hormone called ACTH into your bloodstream. That ACTH travels all the way down to your adrenal glands, which sit on top of your kidneys, and tells them to pump out cortisol, your primary stress hormone. This entire brain-initiated chemical cascade rapidly alters the physical environment of your gut.
How does cortisol actually change the gut, practically speaking?
Well, it has immediate physiological effects. It alters gut motility. It might slow down the stomach, delaying emptying, while simultaneously speeding up the colon, leading to diarrhea. It changes the local secretion of fluids. But if that psychological stress becomes chronic, the changes become much more severe.
The environment actually physically degrades.
Precisely. Chronic stress and elevated cortisol can actually compromise the tight junctions between the cells lining your intestines. This increases intestinal permeability, which is often colloquially referred to as leaky gut.
I've heard that term a lot.
Right. When the barrier becomes permeable, immune cells in the gut lining get triggered, creating low-grade inflammation. This altered inflammatory environment then dynamically shifts the composition of your microbiome, favoring different bacterial species. Furthermore, that chronic stress literally sensitizes those millions of enteric neurons, making them hyperreactive. Normal, everyday digestion suddenly feels painful.
So the brain's stress response physically breaks down the gut environment. But because it's a loop, those physical changes, the inflammation, the altered microbes, the sensitive nerves, they then send distress signals back up the vagus nerve to the brain.
Exactly. The brain receives those inflammatory and neural signals, interprets them as a physiological threat, and ramps up the baseline anxiety even further. The loop feeds itself.
But the beauty of a bidirectional loop is that you can intervene on either side, right? If the highway runs both ways, you could use one system to heal the other.
And this is where some of the most exciting clinical interventions are happening. We now have excellent evidence that targeted psychological interventions, specifically cognitive behavioral therapy, CBT, and gut-directed hypnotherapy, actually yield significant improvements in the physiological symptoms of IBS.
That still blows my mind. You can essentially use talk therapy or hypnosis to physically normalize your bowel habits.
In cases of functional disorders driven by this axis, yes. By calming the central nervous system and downregulating the HPA axis, you remove the stress signals bombarding the gut, allowing the enteric nervous system to reset. And conversely, interventions that target gut function like specific dietary changes can yield improvements in psychological symptoms.
Which perfectly tees up what is unquestionably the hottest, most exciting, and absolutely most controversial area of this entire field, the microbiome and mental health.
It is definitely a controversial area.
We have to be meticulous here about separating the rigorous biological evidence from the speculative marketing claims, because the supplement industry has gone into overdrive on this.
The idea that manipulating the bacteria in your colon could serve as a primary treatment for psychiatric conditions like clinical depression or severe anxiety is captivating.
Oh, yeah?
And that enthusiasm wasn't just invented by marketers. It was ignited by some genuinely astonishing, paradigm-shifting early research in animal models.
We have to dive deep into these animal studies, specifically the germ-free mouse experiments, because the methodology alone sounds like science fiction. Scientists literally rear mice in specialized, completely sterile, hermetically sealed isolators.
It's quite extreme.
Yeah. These animals are delivered via C-section in a sterile environment. Their food and air are completely sterilized, and as a result, they possess absolutely zero gut microbiome. No bacteria, no fungi, nothing.
They're essentially blank biological slates. And studying them allows researchers to isolate exactly what the microbiome actually does by observing what happens when it is entirely absent.
And the behavioral results are wild. A mouse without a microbiome does not act like a normal mouse.
The neurological and behavioral abnormalities are profound. Germ-free mice exhibit highly exaggerated physiological stress responses. When you subject them to mild stress, their HPA axis goes into overdrive, pumping out massive amounts of stress hormones compared to normal mice.
Wow.
They display profound deficits in social behavior, often avoiding interaction with other mice. Interestingly, in some specific behavioral tests, they actually show reduced anxiety-like behaviors. They take more risks. But the overarching picture is one of fundamentally altered, abnormal neurodevelopment.
But the fix is what put this field on the map.
Yeah.
Because these behavioral deficits aren't permanent genetic flaws, they can essentially fix these mice by giving them specific bacteria or by performing a fecal transplant from a normal, healthy mouse. The moment that sterile gut is populated with a microbiome, the mouse's behavior and stress responses begin to normalize.
It is a stunning proof of concept. It definitively proves that the presence of a complex gut microbiome is causally required for the normal development of the brain and the regulation of behavior in these mammals. The microbes are pulling the strings.
And there was one specific study from 2011 that I think illustrates the mechanics of this better than anything else. The Lactobacillus rhamnosus study.
Yes, conducted by researchers at University College Cork, which is a massive hub for this kind of research. This study was a landmark moment for the gut-brain axis.
So instead of using germ-free mice, they took normal, healthy mice. And for a month, they fed half of these mice a specific strain of bacteria Lactobacillus rhamnosus.
The behavioral outcomes were measurable and significant. The mice fed the lactobacillus strain demonstrated significantly reduced anxiety-like and depression-like behaviors in standardized testing mazes, and they showed a blunted stress hormone response compared to the control group.
They essentially gave these mice a probiotic antidepressant, and it worked.
Exactly.
But the detail that elevates this from a cool observation to a definitive proof of mechanism is what they did next. To prove exactly how the bacteria in the gut were changing the behavior of the brain, the researchers performed a surgical procedure called a vagotomy. They literally went in and severed the vagus nerve in a subset of the mice.
It is an incredibly elegant experimental design. When they severed that primary communication cable, the psychological benefits of the lactobacillus completely disappeared.
Because they literally cut the wire.
Precisely. The bacteria were still alive and thriving in the gut, producing whatever metabolites they produce. But without the intact vagus nerve acting as the transmission wire, the brain never received the signal. It proved not only that specific gut microbes can alter behavior, but that the vagus nerve is a required primary conduit for that behavioral effect.
Now, if you were listening to this, your immediate thought is probably exactly what mine was. Sure. Give me a bottle of the Lactobacillus rhamnosus. If it acts as a stress reliever in an anxious mouse, it should cure my anxiety, right? But here we crash headfirst into the human translation problem. A human being is not just a giant bipedal mouse.
We are infinitely more complex. The neurobiology, the diet, the genetics, and the environmental exposures of a human being living in the modern world cannot be replicated in a sterile mouse cage. And unfortunately, the translation of these miraculous animal findings into human clinical success has been incredibly murky.
We do have a lot of observational data in humans, though. We know for a fact that if you take a large group of people diagnosed with major depressive disorder and you sequence their microbiomes and you compare them to a group of people without depression, their microbiomes look consistently different. Certain bacterial species are depleted, others are overgrown.
The observational link is robustly documented, but we immediately run back into the ultimate scientific trap correlation versus causation. What are the confounding factors?
The confounds are everywhere. When we observe that a depressed person has an altered microbiome, the multi-million-dollar question is, did the altered microbiome cause the depression? Or did the massive lifestyle changes associated with depression cause the microbiome to shift?
Think carefully about the behavioral reality of clinical depression. When someone is severely depressed, their dietary patterns almost always change. They might lose their appetite entirely, or they might shift toward easily accessible, highly processed comfort foods.
Sure.
Their sleep architecture is profoundly disrupted. Their physical activity levels plummet. And crucially, they're very likely taking psychiatric medications like antidepressants.
And we know that SSRIs and other medications have a massive, direct antimicrobial effect on the gut bacteria.
Exactly. Every single one of those factors, diet, sleep, movement, medication, exerts a rapid and profound shaping force on the microbiome ecosystem. Therefore, observational studies absolutely cannot prove that the bacteria are the root cause of the depression. They are just as likely to be a downstream symptom of the depressed lifestyle.
Which brings us to the ultimate test: clinical trials on actual humans. If I walk into a health food store, I see rows of incredibly expensive probiotic capsules marketed as mood boosters and psychobiotics. If I believe the marketing on the side of the bottle, this science is totally solved. Are you saying that all of this is basically biological marketing fiction right now?
I want to provide a highly calibrated answer to that because it is nuanced.
Okay.
The underlying physiological mechanism is real. The gut-brain axis is established biological fact. And scientific consensus strongly supports the idea that the microbiome's chemical output clinically influences human brain function and mental health outcomes. But, and this is a massive caveat, the specific commercial claims that purchasing a $50 bottle of Strain X will cure your anxiety or alleviate your depression are vastly overblown and unsupported by the current clinical data.
Because when we actually run the human trials, the results just aren't there yet.
Correct. When you look at the randomized controlled trials, the RCTs, which are the absolute gold standard of medical evidence, where researchers give human subjects probiotics to treat anxiety or depression, the results are incredibly mixed and frustratingly inconsistent.
Really?
Yeah. A few trials show modest, statistically significant positive effects, but many other highly rigorous trials show absolutely no effect whatsoever compared to a placebo.
Why is it so messy?
Because of the ecological complexity we discussed earlier, the studies use wildly different bacterial strains, they use different dosages. The baseline microbiomes of the human participants are all completely different, meaning the soil they are dropping the probiotics into is highly variable.
Right. Everyone's starting from a different place.
Exactly. The science is methodically crawling, trying to untangle all these variables, while the commercial supplement industry is sprinting ahead, eager to monetize the hype. We simply do not yet have the scientific precision to make strong, specific medical recommendations for psychiatric probiotics in humans.
So, if popping an expensive designer probiotic pill isn't the magic psychiatric bullet that TikTok claims it is, what does the robust clinical evidence actually support? What can we do today to optimize this system? The answer is incredibly empowering, but it also feels like a spoiler alert. The winning combination is something your grandmother could have told you 50 years ago.
It is remarkably beautifully straightforward. When you strip away all the hype and look purely at the most consistent, rigorously validated findings in all of the nutritional psychiatry and gut-brain research, it all points to one specific dietary pattern.
And what's that?
Diets that are rich in dietary fiber, high in fermented foods, and characterized by a massive diversity of whole plants are consistently, undeniably linked to both greater microbiome diversity and significantly better markers of psychological well-being.
The absolute poster child for this is the Mediterranean diet. We hear about the Mediterranean diet constantly in the context of cardiovascular health or longevity. It's built around high consumption of vegetables, fruits, legumes, whole grains, nuts, fish, and copious amounts of extra virgin olive oil. But it turns out it is also a profoundly effective brain-protecting diet.
Very much so. Multiple massive, long-term observational cohort studies have repeatedly associated adherence to a Mediterranean dietary pattern with significantly lower rates of depression and reduced risk of age-related cognitive decline. And we now understand, mechanistically, that this neurological protection is heavily mediated by how that specific diet feeds and shapes the gut microbiome.
But again, observation is just observing. Intervention is where you prove it, and that is why the SMILES trial is such a legendary piece of research in this space.
The SMILES trial fundamentally changed the landscape of nutritional psychiatry. It was conducted by Professor Felice Jacka and her team at Deakin University in Australia.
What makes this trial so monumental is that it was one of the first rigorous randomized controlled trials to test a dietary intervention, specifically as a treatment for existing clinical depression. They didn't just ask healthy people what they ate. They took adults who were already diagnosed with moderate to severe clinical depression, and they actively changed their diets.
Yes, they randomized the participants into two groups. The intervention group received intensive dietary support to transition them onto a modified Mediterranean diet, often called the ModiMed diet. This meant drastically increasing their intake of whole grains, vegetables, fruits, legumes, and olive oil, while severely restricting ultra-processed foods, refined sugars, and fried foods. This intervention lasted for 12 weeks.
But the design of the control group is what makes this so scientifically tight.
Exactly. In behavioral trials, if you just give one group a lot of attention and dietary counseling, and the other group gets nothing, the intervention group might improve simply because they are receiving human interaction and care.
Right, a placebo effect from just being cared for.
Yes. So Jacka's team utilized a befriending protocol for the control group. They received the exact same amount of time, attention, and social support from trained personnel as the diet group, but without any dietary advice. It controlled for the therapeutic effect of social support.
After 12 weeks, the results were undeniable.
The dietary intervention group demonstrated significantly greater improvements in their clinical depression scores, measured by the standardized MDR scale compared to the social support group. Over 30% of the individuals in the dietary intervention group actually achieved full clinical remission of their depression, compared to only 8% in the control group.
A third of the patients basically ate their way out of clinical depression in 12 weeks. That is incredible.
Now, we must maintain our scientific rigor. The trial was relatively small, with about 67 completers, and these findings are considered preliminary in the vast landscape of psychiatric research. Diet is not a replacement for psychiatric medication or therapy for severe disease.
Of course.
But the SMILES trial established something vital: the feasibility and efficacy of using dietary intervention as a targeted treatment for depression. It proved that the diet microbiome mental health pathway is not just a theory. It is a plausible, highly actionable clinical target.
So we have clinical proof that it works. Now let's break down the mechanics of why and how it works. We have to talk about fiber. I think most of us grew up viewing fiber purely mechanically. We think of it as biological plumbing maintenance.
Yeah, roughage.
Exactly. You eat fiber to add bulk and keep the digestive trains running on time. But you're saying fiber is actually the primary food source we are throwing down into the microbiome ecosystem to fuel the production of brain-protecting chemicals.
That is the exact biological reality. Human beings lack the digestive enzymes required to break down complex dietary fibers. So when you eat a fibrous plant like a leek, an asparagus spear, or a bowl of oats, that fiber passes entirely intact through your stomach and small intestine.
It survives all the acid.
Yes. It isn't digested until it reaches the dense microbial jungle of the colon. There, it becomes a feast for specific species of bacteria. They metabolize it through a process of fermentation.
And the chemical byproduct of that microbial fermentation is a class of molecules called short-chain fatty acids.
Correct. And the most critically important of these short-chain fatty acids, especially concerning the gut-brain axis, is a molecule called butyrate.
We have to dive deep into butyrate because it seems to be the MVP molecule of the entire system.
Butyrate is incredibly versatile and powerful. Locally, within the gut, it serves as the primary energy source for your colonocytes, the human cells that line your colon. It literally feeds the barrier.
Wow, okay.
By keeping those cells healthy, butyrate strengthens the tight junctions of the intestinal wall, actively preventing that inflammatory, leaky gut scenario we discussed. It also binds to specific receptors on local immune cells, powerfully dialing down localized inflammation.
But butyrate doesn't just stay in the gut.
No, it enters the systemic blood circulation, and once in the bloodstream, it exerts profound systemic effects. Research suggests butyrate can cross the blood-brain barrier.
Wait, really? It goes straight to the brain?
It does. In the brain, it acts as an epigenetic regulator, specifically as a histone deacetylase inhibitor, which is a complex way of saying it can actually alter gene expression in the brain, promoting the production of neurotrophic factors that support neuroplasticity and the growth of new neural connections.
That is wild.
So dietary fiber is not just plumbing. It is literally the raw material your microbes use to manufacture systemic neuroprotection.
And then, alongside the fiber, you have fermented foods, things like yogurt, kefir, kimchi, sauerkraut, and kombucha. When we eat these, we aren't just feeding the existing microbes, we are actively dropping live microbial cultures into the system.
Exactly. These live cultures can transiently and occasionally persistently integrate into your gut microbiome. But even if they only pass through, their presence alters the chemical environment, adds to the metabolic output of the community, and has been shown in recent trials to actively increase overall microbiome diversity and decrease systemic markers of inflammation.
The ultimate takeaway from all this dietary research is actually incredibly relieving. We're talking about insanely complex biology, 500 million independent neurons, 38 trillion interacting microbes, vagus nerve signaling, blood-brain barrier crossing short-chain fatty acids. But the current, well-supported clinical solution is wonderfully simple. You do not need an incredibly complex, dramatic, highly restrictive, or expensive diet to optimize your gut-brain
But no,
axis.
you truly do not. The practical dietary interventions supported by microbiome science are perfectly aligned with the boring foundational nutritional guidance we have had for decades regarding heart disease and metabolic health.
Right, eat your veggies.
Yes. You optimize the system through high dietary diversity eating dozens of different types of plants every week to feed a diverse array of microbes. High fiber intake to fuel butyrate production, regular consumption of fermented foods, and drastically limiting ultra-processed foods, emulsifiers, and refined sugars, which tend to starve the beneficial microbes and promote an inflammatory environment. That simple framework is the foundation of gut-brain health.
Okay, but what happens when the baseline is totally destroyed? What if a conservative dietary shift isn't enough because the ecosystem has been decimated? This brings us to the most dramatic nuclear option in all of microbiome science, fecal microbiota transplantation, or FMT.
It is the most direct, unequivocal, and frankly astonishing demonstration that the microbiome is causally relevant to human health outcomes.
Fair warning to you, if you aren't familiar with this, the process is exactly what it sounds like. FMT involves taking fecal material from a rigorously screened, healthy, diverse human donor, processing it, and transferring it directly into the gastrointestinal tract of a sick recipient. The goal is to literally reset or entirely replace their dysfunctional microbiome with a thriving, healthy one.
The visceral reaction to the concept is totally understandable, but its clinical success in one very specific, life-threatening application is an undeniable triumph of modern medicine. The most established FDA-approved application for FMT is in the treatment of recurrent Clostridioides difficile infection, universally known as C. diff.
And C. diff is an absolute nightmare scenario. It is a highly aggressive bacterial infection that causes severe, relentless, life-threatening diarrhea and massive intestinal inflammation.
And the pathophysiology of C. diff beautifully, tragically illustrates the ecological nature of the gut. C. diff infections almost always take hold after a patient has been treated with heavy, broad-spectrum courses of antibiotics for a different issue. The antibiotics do their job, but as collateral damage, they wipe out the normal, healthy, diverse gut microbiota. They essentially clear-cut the biological forest.
And when the forest is clear-cut, the weeds take over.
Exactly. The C. diff bacteria, which naturally forms incredibly hardy spores, might have just been lingering harmlessly in the gut in very small numbers, kept in check by the millions of other microbes competing for nutrients and space. But suddenly, with the native ecosystem annihilated by antibiotics, C. diff has zero competition. It rapidly germinates and colonizes the empty, depleted gut, pumping out toxins that destroy the intestinal lining.
And trying to treat C. diff with more traditional antibiotics often fails, right?
Yeah.
And it leads to a recurrent infection because you're just dropping more bombs on an already clear-cut forest. You never let the natural ecosystem recover. But FMT takes a totally different ecological approach. You take the healthy donor fecal matter, which is a dense, highly competitive, fully functional ecosystem, and you introduce it into the patient's colon, usually via colonoscopy or specialized capsules.
The goal is rapid ecological restoration. You are replanting the forest, and when you reintroduce that massive diversity of healthy microbes, they immediately begin competing for space and nutrients. They simply outcompete and suppress the C. diff.
And the clinical success rate is staggering.
It is nothing short of a medical marvel. In multiple rigorous randomized controlled trials, FMT resolves recurrent C. diff infections, with a success rate consistently hovering around 80 to 90%.
Wow, 80 to 90%.
Yes. In the context of modern medicine, especially for a severe recurrent condition where all standard pharmacological alternative treatments frequently fail, an 80% curative success rate is extraordinary.
So here's the obvious pointed question that everyone asks when they hear that statistic. If this works so miraculously for C. diff, 80% is an insane success rate in medicine, why can't we just pop a donor pill to cure depression or metabolic syndrome tomorrow? What's the holdup?
If we connect this to the bigger picture, we have to return to the core concept of ecological complexity. With a C. diff infection, you are dealing with a relatively straightforward ecological task. You are replanting a clear-cut forest to crowd out one single specific opportunistic weed.
Right.
But conditions like inflammatory bowel disease, metabolic syndrome, autoimmune conditions, or clinical depression are infinitely more complex, and they involve deeply entrenched feedback loops with the host's own biology.
So researchers are actively trying to use FMT for these conditions, though.
Oh, it is an incredibly active and well-funded frontier of clinical research. But the results so far highlight the immense difficulty of the task. For inflammatory bowel disease, specifically ulcerative colitis, FMT shows promise for some specific subtypes of patients, but it requires highly specific superdonors and often multiple continuous transplants. For metabolic conditions like obesity or insulin resistance, the results have been highly mixed and often transient. And for psychiatric applications like treating depression or autism spectrum disorder via FMT, we were still in the very early, highly experimental stages.
Because it's not just about the seeds, it's about the soil. You can't just take a bunch of exotic plants, drop them into a completely different ecosystem, and expect them to thrive if the climate is wrong.
That is the exact limiting factor. You can introduce a perfectly healthy, diverse microbiome into a recipient via FMT. But if that recipient's underlying host genetics, their ongoing highly processed diet, their baseline chronic stress levels, or their hyperactive immune system are still creating a highly inflammatory, inhospitable environment, well, those newly transplanted microbes will not engraft.
They just die off.
They will not survive. They will simply wash out of the system within a few weeks, and the patient's original dysfunctional microbiome will reestablish itself. The microbiome is a deeply complex, interacting community that is inextricably linked to the host. This immense ecological complexity is why manipulating it for complex systemic diseases is so hard to predict, and why translating a miraculous C. diff cure into a depression cure is going to take decades of meticulous science.
That makes total sense. The biological soil has to be ready to accept and nourish the seeds, which naturally makes me wonder, when is the original soil laid down? When did we get our very first seeds? This brings us to a massive component of our health trajectory, the microbiome in early life. This is when the foundation of the second brain is actually built.
This represents a critical, incredibly sensitive developmental window. The human gut microbiome is essentially assembled and established in the first 2 to 3 years of life, and the specific conditions of its establishment have profound, lasting consequences for our lifelong immune and metabolic health.
Because the starting line is basically a blank slate, right? The infant gut inside the womb is largely, if not entirely, sterile. The massive foundational colonization event happens the moment they enter the world. And this is why the mode of birth matters immensely.
Birth is the first major microbial exposure. During a vaginal delivery, the infant passes through the birth canal and is extensively coated in the mother's vaginal and fecal microbiota. From an evolutionary perspective, these are the exact specific microbes that have historically colonized the human gut for millennia. They are the pioneer species.
But a cesarean section bypasses that specific exposure entirely.
Yes. A C-section drastically alters the initial colonization protocol. The infant is not exposed to the maternal, vaginal, or fecal microbes. Instead, they are colonized primarily by the microbes found on the mother's skin, the hands of the medical staff, and the ambient hospital environment.
And this isn't just a temporary delay where they catch up a week later. It creates long-lasting structural differences in the ecosystem.
The differences in those founding pioneer microbial populations can persist for years, and massive observational studies consistently associate these early developmental differences with variations in immune system calibration, noting a statistically increased risk for developing allergies, asthma, and potentially other immune-mediated health outcomes later in childhood for infants born via C-section.
This feeds directly into a concept that I find absolutely fascinating, and it totally reframes how we view dirt and germs. The old friends hypothesis. This was proposed by a microbiologist named Graham Rook, and it's essentially a highly refined, modern update to what we used to colloquially call the hygiene hypothesis.
Yes, the original hygiene hypothesis, which emerged in the late 1980s, broadly suggested that our modern environments were simply too clean. The lack of childhood infections was theorized to be leading to the massive rise in allergies and autoimmune diseases we see in industrialized societies. But Graham Rook refined this beautifully by focusing on evolutionary biology.
It's not just about getting sick with random dirt or random viruses. It is specifically about our evolutionary partners. We evolved over millions of years alongside specific categories of harmless organisms, microbes in the soil, in our food, and in our guts. These are our old friends. And our immune systems didn't just tolerate them, they evolved to actually expect to encounter them in early life.
That expectation is the crucial mechanism. The human immune system is an incredibly powerful, lethal defense network. But when a baby is born, that system is largely untrained. It needs to be calibrated.
Right.
It requires early life exposure to these specific old friends so it can learn to distinguish between what is a harmless environmental passenger, what is a beneficial symbiotic friend, and what is an actual deadly pathogenic threat.
To get a bit technical, this is about training specific immune cells, right? Regulatory T cells.
Exactly. Exposure to these diverse, harmless microbes stimulates the development of regulatory T cells, or Tregs. You can think of Tregs as the brakes of the immune system. They promote tolerance. They tell the immune system to stand down when it encounters harmless things like pollen or peanut protein or our own human tissues.
So if we use an analogy, it's exactly like training a highly aggressive guard dog. If a guard dog never sees a mailman as a puppy, it grows up isolated, and it thinks the mailman is a deadly threat later. Our immune system is the guard dog, and we've basically stopped letting it see the mailman.
That is a brilliant, highly accurate analogy, and the industrialized world has systematically removed the mailman in multiple compounding ways. We have incredibly high rates of antibiotic use in early life and infancy, which act like bleach on the developing microbiome, inadvertently wiping out these vital early exposures.
Yeah.
We have a massive shift toward urban living, resulting in vastly reduced contact with diverse environmental microbiomes in soil, vegetation, and animals. We have altered infant feeding practices. All of these factors combined remove the old friends.
And the guard dog gets confused.
Consequently, the immune system, the poorly trained guard dog, fails to develop sufficient regulatory brakes. It becomes hyper-reactive, directly driving up the prevalence of immune-mediated conditions, allergies, asthma, and autoimmune diseases.
So this early life window, the first 1,000 days, is a critical developmental period. Just like if a child doesn't hear human language in the first few years of life, the neural pathways for language close, and they will struggle to ever learn it perfectly. If the immune system doesn't see these microbes early on, it struggles to ever calibrate properly later.
The microbiological window is functionally identical to the critical periods for cognitive and language development in the brain, which is exactly why the proactive interventions suggested by this branch of science are so incredibly important for public health.
And these interventions aren't about buying expensive infant probiotics. They're foundational lifestyle choices. It means supporting vaginal births whenever they are medically safe and possible for the mother. It means strongly encouraging and supporting breastfeeding, because breast milk doesn't just provide human nutrition, right?
Right.
It contains highly specific complex sugars, human milk oligosaccharides, that exist solely to feed the infant's developing bifidobacteria. It means being highly judicious and avoiding unnecessary broad-spectrum antibiotics in infants and toddlers unless absolutely medically necessary. And honestly, it means letting kids go outside, get messy, and play in the dirt to promote massive, diverse microbial contact.
Precisely. We need to actively foster that early ecological diversity to build a resilient, well-calibrated immunological and neurological foundation for the rest of their lives.
Wow. Okay, we have covered an immense amount of biological ground today, from 19th century dog experiments to the HPA axis to germ-free mice and fecal transplants. Let's pull this all together for you in a clear synthesis. If you take away one thing from this deep dive, let it be this. The gut-brain axis is not a fleeting wellness fad. It is a heavily verified, deeply integrated, massively complex biological system. Your gut and your brain are in constant bidirectional communication via a massive neural network and the chemical output of 38 trillion microbes. And the choices you make every day, your diet, your lifestyle, your stress management, profoundly and directly influence your mental and physical health through this exact pathway.
And remember, while the biology is extraordinarily complex, the practical application is highly accessible. You absolutely do not need expensive, hyped-up, heavily marketed supplements to manage this system.
Great point.
The most evidence-backed interventions are foundational and available to almost everyone. Eat a diverse plant-based diet rich in fiber to fuel butyrate production. Incorporate fermented foods. Avoid unnecessary antibiotics. And actively manage your chronic psychological stress, because the science definitively shows that your mind changes your gut environment just as much as your gut changes your mind.
It is wonderfully empowering to realize you have that much control over your internal ecosystem.
Yeah.
But I want to leave you with a final lingering thought to mull over, a slightly philosophical question based on the sheer scale of the science we've unpacked today. We inherently tend to think of our thoughts, our moods, our stress resilience, and even our daily cravings as the absolute core of our independent human identity. It's our mind. But if your daily anxiety levels, your emotional resilience, and even what you intrinsically desire to eat for lunch are being chemically heavily influenced by a colony of 38 trillion non-human microbes, an ecosystem that you inherited at birth that shifts with every meal and that vastly outnumbers your own human cells.
Where does the biological you actually end and the ecosystem begin? A fascinating, unsettling, and brilliant question to leave it on.
Thanks for diving deep with us.